What we think of an orchid is that it encompasses many orchids in this family, but there's a lot of them that are much less showy and much less obvious than what you're probably familiar with. The flowers are usually perfect, so they're bisexual, and they're going to have a really weird anatomy to their reproductive parts in the flowers. So we're going to spend some time talking about that because it's a shear drive trait for the group. They're definitely zygomorphic though, because they're not going to be able to have more than one plane of symmetry where you can cut it this way, get a mirror image, but you can't do it this way. And that's because oftentimes one of the petals looks very different, and one of the sepals looks very different than the rest of the sepals. Range of size and showiness, obviously the big ones we're familiar with are the really showy ones, which are going to be similar to the ones we're going to look at today. But you can find some that have much smaller flowers, which is going to be one that we're going to learn in lab as an example. I've seen ones that have flowers the size of the tip of my pinky. They can be white, they can be pink and colorful purple, they can be kind of bronzish colored. That's a real common one for orchids, greenish and yellow, and you may not even notice them for that reason. So it's a big family. The flowers are going to have 6 tepals, and I will say that this doesn't necessarily in this case mean that they all look exactly the same. What this is talking about is it means that all of them are going to look like petals. They're going to have that texture and consistency of petals, and they're going to be somewhat showy in that case. So this, for example, you can see there's one sepal here, sepal here, and we've got petal, and then this is the weird petal. We're going to talk about that one in more detail. That would make up the 6 total tepals that we have. They're variable in terms of their shape and color, but one of the tepals, and this is going to be actually one of the petals, is referred to as the labellum. This is the labellum right here. Okay? It is kind of sculptured in the sense that it doesn't lay nice and flat like you expect a petal to. All of these are kind of nice and flat petals and sepals like we expect them to be. With this one, you can see it kind of curves. It kind of has some 3 dimensions to it. That's what I mean by sculpture. And it serves an important function in orchids because it's essentially the landing platform for pollinators. So when they come in, whatever the pollinator happens to be, so for example, if it's a type of bee, it would come in and that's what it would land on to access this weird reproductive structure that we're going to look at right here in more detail. So the labellum, this is going to be definitely something you're going to want to know for the test. This is something I would consider a unique characteristic for this family, so it's a term you absolutely want to know, well enough that you could write it out. And be able to recognize which one of them is the labellum. It's going to be one of the petals, and it's the one that's going to look kind of sculptured and decorative. And we'll come back to this in a second. Then we have our calyx, where sometimes the middle sepal is going to be different from the 2 that are on the sides. So you can see in this orchid right here, these 2 linear ones that stick down, those are actually lateral sepals, the 2 side sepals. And then you've got this one at the top right here, that's the dorsal sepal that looks very different than these 2 down here. Whereas the previous picture I showed you, all the sepals look the same. So it just depends on the species. In regards to our corolla, sometimes it's spurred. Now I've mentioned spur multiple times in lab because we've had 2 plants so far that have that. The spur is something that's you can find in violets, or in the case of our lab plant, it was the field pansy, which is essentially a sac that forms on the back side of the flower from the petals. And for the viola that we learned, it wasn't real big. There's other species that have bigger ones. And for those of you that were in the Tuesday lab, we learned about another one out in the field when we went out to our site over by the UTM farm that was called the yellow fumort, and that one had a spur as well. And what was characteristic about that one is it didn't stick straight out like this, it actually recurved, which meant it pointed downward. So these will sometimes have a spur in the back of them, which is a sac-like projection from the flower parts. And it's usually the same color as the petals themselves. One of the petals, of course, is the labellum. And you can kind of see how different it can look in terms of its sculpturing. So for example, this one right here is the labellum. So you can see there's these things that stick up on this one, which makes it more sculptured, 3-dimensional. This one, it looks kind of like a slipper. We'll get to that one in a little bit. So it's a big kind of almost inflated sac. That's actually the labellum there. And this one is just real feathery and much more decorative looking than the other ones up here. Now let's talk about the reproductive structures. We're going to discuss this in 2 different slides, one that deals with the androecium, one that deals with the gynoecium. But what I want you to know first about them is that the androecium and the gynoecium are technically fused together. Okay, so in some ways it is one structure, but you have your male parts of it and you have your female parts of it. Usually there's only one stamen, and it's actually fused to the style and stigma. When it does this, it forms essentially a column, a structure in the center of the flower right here. That's referred to as the gynostegium. It's also sometimes referred to a column. These two terms mean the same thing. So if on the test you want to write down gynostegium, that's fine. If you want to write down a column, that's fine as well. They mean the same thing. But this is an important one because this is a shared derived trait for the orchids, meaning this is the only family that has this. It is a defining feature for them, and every species that's in this family is going to have this gynostegium. Okay, and so this is kind of one that they've pulled out, so you can actually see the column as a whole. They've actually pulled it out of the flower. When we feel it in the flowers that we're going to look at today, it'll feel kind of solid. It's not kind of nice and flexible the way the petals are. You'll be able to feel the solidness of the column. So that's the gynostegium right there. And the cap, or the anther, is actually kind of like a cap. And you'll see how it's kind of popped off here. This is the cap anther right here. When that pops off, underneath it, you're going to see these two weird little yellow balls. We're going to get to that in a second. Those two little yellow balls are what are referred to as pollinia. They are not pollen grains, they are whole collections of pollen grains. In other words, it's not an individual pollen grain. It is a whole bunch of pollen grains all grouped together, kind of glued together in a waxy mass. So you've got one on this side, and you've got one on this side. Each one of those is called a pollinia. Now the advantage of this is that those are essentially going to get transported as a whole. So you don't have separate little pollen grains going to each of the flowers that they're going to pollinate. You have a whole cluster, a whole pollinia that's going to get transferred to a flower. There's a reason for that too, because we're going to find out there's a lot of ovules in orchid flowers. So there's a lot of things that need to be fertilized. So you need a lot of sperm, all delivered at the same time. One pollen grain is not going to do it. So bring it all in mass. Now I will tell you that there's some additional features that are going to be involved in this structure. The two yellow balls are the actual pollinia. You can see that they're sitting on top of a stalk right here. That's kind of sticky. We'll get to that in a second. If you talk about the whole thing right here, this is called a pollinaria. We'll get to that in a second. But the two actual waxy balls of pollen are the pollinia. All right, now the gynoecium is going to have an inferior ovary. So if you go down to the base of the flower, here's actually where all the sepals and petals are attaching right here, and our ovary is actually down here below that, so it is in fact an inferior ovary. It's kind of hard to figure that out though, because you got this weird column that's going to take him sticking up in the middle of it, so you might think, well, if the ovary is somewhere in here, then it's actually above where the petals are, but that's actually not the case. This is the style stigma portion, and the anthers portion. Down underneath where the flowers are is actually where the ovary is. Okay, so it's inferior. It has 3 fused carpels. And inside of those carpels, there are millions of ovules. So it has the capabilities of producing millions of seeds. And that's why it's so important for that whole mass of pollen grains to come at once to this, so that all of those ovules, or many of them, can actually get fertilized and form into seeds. They're going to be really tiny though, because there's so many of them. Essentially, when one of these capsules opens up, it looks like pepper, little pepper granules. The style is fused to the androecium, and it is a major component of the gynostegium. So most of the tissue, most of the mass of that column is actually style material. Now, if you look at the column up close here, okay, so we've got all of our style region down here. Here's your anther cap right up here underneath that. You can kind of see through it and see where the pollinia are. Right, this is the sticky stalk facade to that. We'll get into that in a second. But you'll see right under here, there's a chamber, kind of like a little cave that's in there. That is actually the stigmatic region. So when the pollinator comes to this flower, this pollinia, which is going to be stuck to it, right, is actually going to have to get inserted into this cavity right here for pollination to occur. We're going to do that today. So we're going to play with this so we've actually get a chance to see what this looks like. Okay, so here's another close-up of what it looks like. Okay, so we've got our side view, our front view of our flower. You can see where the column is, the gynostegium, the pollinia are inside here. And if we look closely, kind of zoomed in on part of the flower right here, this sculpture, see these two sides right here? That looks like they should be separate petals, they're not. That's all part of one petal, which is also a part of this. So this whole thing plus these things are all one petal, that's the labellum. And then you have these bigger ones on the side that are the other types of petals. And inside of there, you can see sticking up, that's gonna be our Anastasia. Now let's talk about the pollination stuff and the seeds. Because we've got so many ovules in these fruits, you can see how tiny the seeds are when they actually release, and it looks like pepper grains when you're shaking pepper out of it. These are actually capsules, and so you'll see they'll split along different suture lines here. They're multicarpellate, as capsules should be. There's 3 carpels. And if you look closely at the seeds, oftentimes they're winged. They have these little translucent wings on either side of them. So they're tiny, and they also have wings, so they disperse pretty well in the wind. So let's talk about the pollination mechanism here. The actual unit that gets transferred is not a single pollen grain or single pollen grains. It is this entire thing that's called the pollinarium. Right? The pollinarium is made up of multiple things. It's made up of the 2 pollinia. So just the yellow balls are the pollinia, those waxy balls of pollen right there, plus this sticky stalk down here that's made from the anther and from the stigma. It is very sticky. And it will stick to the back of bees and other pollinators that come to the plant. And so when they come into this other flower, this portion right here, this whole thing, is going to get deposited into that stigmatic area. Now the adaptive significance of this is it ensures fertilization of a large number of ovules with just one visit. You don't, if you've got millions of ovules in here, you don't want to have a bee come to that a million times to make sure that all of those ovules are fertilized. This ensures that most of them, if not all of them, are going to get fertilized in just one visit from that bee. So a significant advantage. Now let's get some up close and personal views of what this looks like. So I'm going to have groups come up in groups of 3. I've got 3 different plants here. Everybody's going to get a chance to do this. And so you should be very familiar with the anatomy of the orchid flower. Hint. Okay? Make sure you're very familiar with the anatomy of the orchid flower and how the pollination works. Okay. I also, when you come up, want you to feel the leaves. One of the things I told you in the last class about the leaves of orchids is they tend to be coriaceous, which means they feel kind of leathery and thickened. So make sure you get a chance to feel what those feel like. All right. I kept these closed up because otherwise they tend to fall off in transit. For any particular color, I was going for ones that had lots of flowers on them. So we've got enough for everybody. Here it is. All right. So I think I'm going to start on this side of the room. I want groups of 3. So I've had 3 of you go ahead and come up to the front. The rest of you can talk amongst yourselves, or you can watch what they're doing because you're going to have to do this again here yourself. Make sure you feel the leaves. Pick an orchid, doesn't matter which one. Each of you is going to get one of these. I'm going to give you this one, but when you touch the flower, go ahead and just kind of hold it like that. Okay. All right. So what we're looking for here is we're looking first for our Anastasia. So go into the flower, into the center of the flower here. First, point to you where the labellum is. Yes, and it's all of this. It's this, this and this all together, that's all one pedal right there. All of that, that's our labellum. Now in the center here, this is your column or your diastadium. So you've got your labellum. That's your diastadium. So go ahead and feel that. Pinch your diastadium and feel what it feels like. You feel pretty solid. That's okay, we're gonna do that anyway. Alright, so what your pointer there is for exactly what you just did. You see here, this is your antler cap on top of your diastadium. You see that little pointed triangle sticking down there?
What? Nothing.
You wanna get this right underneath that, lift it up, and pull that cap. If you can find that triangle,
Who is?
flip that off.
What'd you say, Jay?
What?
What is it?
Okay.
Definitely not this, so.
Perfect.
Oh.
Okay, yeah, and see how it's sticking to your forceps there?
Huh? I did?
Yeah, I did.
Pull that, and now what you
That is
Did
true.
you really? Yeah. I did.
The bed was too small.
I said get between the crevice wall.
I mean,
That's
that's
like.
like, people were like hanging off the end of it. It was like-
I was tired of waking up to like breath in my ear, like. Alright. Wait, what about?
I heard you had to put yourself in the shower to contain yourself though.
Huh?
Didn't you have to put yourself in the shower to contain yourself?
Yeah.
I got really quiet when I said that. Really weird.
Alright, now for yours, so what you're looking at there, you should see the two polyamble, and you should see this, the sticky stuff, right? Okay, so the
I had a
whole
dream
thing,
about
you've got
conclave
your
last night,
polyamble.
actually.
Really?
Take that.
No. No, we were just like back at that hotel, and we were doing, I don't even know what we were doing. We had like
Probably
a class,
drinking.
we had a class there.
You see
I know.
that? Oh, right there, yeah, that's what I want you to do. Okay.
That's crazy.
It's not because it's very sticky, it's hard to get it off, yes. There it is. And what you guys can do, since you have the same wire pitch, too, you switch. You pollinate this, and you pollinate yours.
I saw
And
him
when you're
working
gonna
in the
put it,
wildlife job.
here's this little
That is what
crazy
your
thought.
degree is gonna be in.
It's a crazy thought.
I know.
You're one of my favorite
Yours.
things.
But like how I went out flying. Like, I really good wildlife job.
All right there.
Too bad.
Yeah, nice, right off.
Seems great. It's just different.
Okay, excellent.
It's like a part-time like
It has
that.
to rub off of it.
Wildlife.
Some of them actually try to copy the flower, that's how they work. All right, next thing.
You have a long way to go.
I'm not good. I have one of the other politics.
Jason, that's not inspired. You should try it.
No, I've worked on politics for a while, but probably not, probably not.
Oh, yeah, it was like mine. Third grade is literally when you get
Like
it.
me, maybe.
Yeah,
I'm getting
Probably a tick bite or a chigger.
and
I'd rather get a tick bite than a chiggers because chiggers are freaking annoying.
I hate to. Dude, I was at the market, I was like,
Oh, boy.
After that really just gets rid of them. You just fed you yesterday.
Thank you, man. Thanks for feeding me yesterday.
My calendar start going to each other with internet when I have all these things. I was not. We don't have a calendar.
You think you passed yesterday? You think you passed yesterday?
Oh yeah, I think I only mixed up. I mixed up the
This is really good though.
Venus and New Glass sticky mouse here chickweed. I think she went over when she went over all of them. I'm pretty sure I got all the others right.
Yeah, there's only two. There's like what, 4 points.
Yeah.
It's still like a A, I think.
Yeah, so that's what we're
Right.
I mentioned it is. This stuff we're going on now. It's
Perfect, perfect. All right, now your antler cap already came off. So you guys need to do is clean off that little white piece that's right on the tip of it.
I can't imagine it's gonna be any more difficult than the other two. Those were like.
The first one was just a typical scared of sword exam though. It's like, if you just like skip the wrong page of notes or something.
Better,
Oh,
perfect.
Studying,
Alright, so all of you, what you have
overanalyze
right now is you have
stuff,
this.
get really
You have a common area.
read the page.
You've got your two common in there, the two wax balls,
It's
and
not
you've got
really
that
what you
sticky
know, it's
stuff
just
down
that
there
you know the
underneath
words
it.
that he said, like,
Now, go to,
well
I'm gonna
enough
have
that you
two
can
pieces
just
of
random arbitrary
Yeah.
facts to put on it. So as long as you study everything, then you're good. Yeah, so
the problem is, I usually miss one.